Hello everyone, welcome to part 9 of this video series. So in this video, we are going to talk about cfDNA, which is a new research topic. So let's get going. So first of all, as a human, you can definitely draw your blood from your hands or from other places. And that blood can be circulated in high velocity using some kind of process called centrifuge. If you apply that process in blood, then your blood is going to be divided into three parts. The upper part is the plasma, the middle part, the thin part is the Buffy coat, and the last part is the erythrocyte. Let's see what each part signifies first. So the plasma is what contains cfDNA. Now cfDNA is not the actual large DNA that you know about, the 3 billion base pair. It is simply fragments, small fragments of DNA floating in the blood. And those fragments can be of length around 100 to 250 base pair. There are obviously longer cfDNA and also shorter cfDNA, but pretty much, 100 to 250 base pair range contains most of the cfDNA. Then we have the Buffy coat. It contains the white blood cells and platelets. So white blood cells are the ones fighting as an immune system for our body. And this is the true genomic DNA. So if you want to get your genomic DNA from any one of these parts of the blood, which is 3 billion base pair long, then this is the part you should be looking at, the white blood cell. Finally, we have the erythrocyte, which contains the red blood cell. The red blood cell is the one that carries the hemoglobin and the oxygen, and it doesn't have any DNA involved. It doesn't have any DNA involved is because it doesn't have any nucleus. Because from part 1 of the series, you know that DNA resides inside the nucleus. So no nucleus, no DNA. We know that there are different types of blood cells, red blood cell, white blood cell, platelet, etc. All these different kind of cells that we know actually generate from the hematopoietic stem cells. And these are found in bone marrow. So I think all of you have heard about bone marrow transplantation, like some people have problems in their bone marrow and then they severely need a transplant, is because without the bone marrow, you cannot really create the different kind of blood cells. And hematopoiesis is basically a process of blood cell formation. Now, I'm talking about hematopoietic cells. There is a reason for this because in the last slide, I am talking about clonal hematopoiesis. So clonal hematopoiesis is a very important topic for cfDNA. So next, let's talk about sources of cfDNA, like how does cfDNA appear in the blood. The first and the very natural process is the apoptosis, because your body is programmed to kill your cells when they are actually very old. It is a natural process, and that is the main source of cfDNA under normal condition. Then you have necrosis, which is uncontrolled cell death, and this happens when you have some kind of injury or inflammation in your tissue. Then we also have fetal DNA. This is very interesting because like when a woman is pregnant and she has a baby in her placenta, then from that baby, some cfDNA, some DNA fragments actually come into the bloodstream of the mother. And these cfDNA are actually very important for something called non-invasive prenatal testing. NIPT. This is a very interesting field of research. And this term, non-invasive, is very, very important because if you want to deal with cfDNA or if you want to analyze based on cfDNA, you don't have to do any operation, you don't have to do any surgery, you don't have to take tissue out. You just need the blood, right? You just need to get some injection, get the blood, and you can do everything. That is why it's called non-invasive. It's not invasive at all. Then you have organ transplant. So if you take some organ from a donor, because your own organ is damaged, right? That is called organ transplant. And if you want to monitor organ transplant rejection, sometimes what happens is the organ that you took from someone else is not being accepted by your body. So how exactly do you monitor that? So you can also do that using cfDNA, the donor-derived cfDNA. And finally, this is, I think, the most crucial topic now in the research of cancer. The latest research of cancer are revolving around this, which is called ctDNA. So ctDNA is called circulating tumor DNA. It is also cfDNA, but it is derived from tumor cells. And this kind of ctDNA actually reflects the genetic and epigenetic alterations of cancer and is the basis for non-invasive cancer detection and monitoring. So normally, if you do want to do these things, like if you want to detect cancer and if you want to monitor the progress of treatment, you need to take tissue through operative or surgical measures. But this is not required if you deal with ctDNA. Just to elaborate a bit more, so this is your original tumor. Say you have a tumor in your liver right? We have liver cancer. So, I mean, those tumor cells are very aggressive. They grow very aggressively and uniformly, and they can actually go inside your bloodstream as well because of those uncontrolled growth. And then from these tumor cells, I mean, since they're ill-formed, DNA is actually degraded and they go into the cell, go into the bloodstream, which is called basically ctDNA. So these blue things are ctDNA right here. So in a cancer patient, the bloodstream is basically a mixture of cell-free DNA and ctDNA. Okay, so now let's see some important properties of cell-free DNA or ctDNA that can enable us to detect cancer or to progress, monitor the cancer. So as I mentioned before, most of the cfDNA fragments are in the range of 100 to 250 base pairs. So if you are a healthy person, like the green one, you can have this green curve as your fragment length distribution. So you can see that there is a peak around like 160, 166 base pair. So your peak will be somewhere around here. So most of the cell-free fragments will belong to 166 base pair if you are a healthy person. And these are just counts. So like around, I think how many zeros are here. Anyway, I think it's 250K. Yeah, 250K fragments are actually there for the 166 base pair fragment length. And yeah, as you have more tumor fractions, so TF means tumor fraction here, there will be a shift in this distribution. So this is very interesting because this can actually enable you to detect cancer and to like monitor tumor progress. This is one of the things. Then you can also use the copy number profile. Now, if you want to know more about copy number profiles, I have a separate video on this. I mean, you can check the entire series. I think it was in part 4 probably. So you can check out that. Now you can see that in a normal cell-free DNA, the coverage profile is pretty flat. But if you have some tumor fraction like 27%, tumor fraction is actually there in this sample, then you will have a lot of like amplifications, deletions, etc. So that is something you can use to also like monitor the progress of cancer patient. Then you also have the mutations. So obviously, if you have cancer, you have some mutations. For example, there are some common mutations for different cancer. For colon cancer, you have the TP53, CRAS, APC, etc. For breast cancer, you have BRCA1, HER2, TP53. And like for all the major cancer types, we know commonly which mutation types are actually there, different in the cancer types. And those mutations can also be seen in the cell-free DNA or ctDNA of those cancer patients. So you have this population, so you have these cancer mutations, the red ones, and you have non-cancer mutations. Now, if you have been following my lecture series, in the previous part, part 8, we talked in detail about the DNA mutation, and we also talked about SNPs, which are called like single nucleotide polymorphism or SNP. So these SNPs are actually natural variants. In humans, they are not related to cancer. So they can also be there. And then you have cancer mutations. So it's a bit challenging, like which ones are actually SNPs and which ones are cancer mutations. You have to sort of understand that through analysis. That is one challenge. Then you also have the methylation. Now we talked about epigenetics and methylation in one of my videos in this series. I think it was in maybe part I don't know, part 3 or 4, something like that. It was right after copy number alteration lecture. So you can check that out. So I mean, healthy control and different cancer types have actually different methylation profiles, and you can sort of analyze those profiles and like understand like what type of cancer you have. That is also another type of approach people take using cell-free DNA. So all these analyses, I mean, they are very good. The most robust ones are actually based on copy number profile and the mutations. They are robust, but they also have issues. So I will talk about those first. So I think the most pressing issue that we have now is the clonal hematopoiesis, also called CH. Let me explain what this is. So in the first slide of this lecture, I talked about hematopoietic cells. So these are cells that are responsible for the production of blood cells. We know that. Now, some hematopoietic stem cell may acquire some genetic mutation. I mean, they don't have to be cancer mutations. They can be any mutation. Obviously, since these hematopoietic cell will keep producing blood cells, so these mutations will actually spread in the blood cells as well, right? And they can be spread in large populations of blood cells, which they call clones. So large population is called clones, basically. That is why it's called clonal hematopoiesis. It usually happens in older adults, like people who are over 60, but then again, cancer patients are also more likely to be old people, right? I mean, it is more probable for older people to get cancer as well. Now, if you are using some mutation callers, we talked about mutation callers in part 8 of the video series. Now, mutation callers don't really understand which mutations are actually from clonal hematopoiesis, like these hematopoietic stem cell mutation, and which ones are actually from the ctDNA, the circulating tumor DNA. So you can falsely detect cancer in people who have clonal hematopoiesis. This is a big problem. And it is not very restricted to mutations only. I mean, it can also be driven by copy number alterations. So there are like CH version of CNAs, copy number alterations. They have a name, also nice name, mosaic chromosomal alteration or mCAs. And they can surely confound or hamper your copy number based analysis of disease diagnosis. So that's all about clonal hematopoiesis. One last important topic I want to talk about here, which is also found in many researches, especially the mutation researches, is the matched normal. So what is a matched normal? So suppose you have a cancer patient. You have a cancer patient. You want to monitor his progress. You want to create cancer. You want to do many analysis on the patient. Now, obviously, cfDNA is a very popular method to do this because it is non-invasive. But we know that in the cfDNA, there are mainly three types of mutations. One is the clonal hematopoiesis. Now let's talk about that later on. Let's forget about that for a second. But we have two other types of mutations. One is a natural mutation, which is basically SNPs or single nucleotide polymorphism (SNP), and the other one is what is known as cancer mutation, right? Coming from circulating tumor DNA. So when you are analyzing the cfDNA of a cancer patient, how do you know which ones are SNPs and which ones are cancer mutations? So a good way to do that is to first extract the matched normal from the patient. So how do you do that? Now, matched normal is something you can actually derive from the Buffy code. So we know that when we centrifuge the blood, the middle portion, you can look at the first slide again, the middle portion, let me also show you here actually, just to clarify the topic. So you have this part, right? This Buffy code part. So this Buffy code part, as I told before, it has a true genomic DNA, right? So this true genomic DNA, it doesn't really have the circulating tumor DNA. It has the original DNA that we have. And that is why the mutations that these DNA have are actually SNPs. So if you record those mutations, then when you analyze cfDNA, and if the mutation caller say tell you that I think that this mutation is a cancer mutation, then you simply check. And then you simply check the Buffy code mutations. So if that mutation is there in the Buffy code, then no, this is not a cancer mutation. This is a natural variation. If that mutation detected by your VAF caller, mutation caller, does not exist in the Buffy code, then you can actually say that yes, this is probably a cancer mutation. That is the thing. That is the function of matched normal. So matched normals are widely used for mutation-based cancer research because of this. But there is a catch. Matched normals can also have clonal hematopoiesis, CH. So it is not free from that problem. So CH will always cause you a headache, and you need to be very wary about these things. So that's it for this tutorial. Hopefully in the next part of the series, I will be talking about phylogenetic trees and evolution. And that's a very important topic if you're working with evolutionary trees and such construction. So hopefully see you in the next tutorial. And if you enjoy this series, make sure to like, subscribe, and you can even donate to me. You can buy me a coffee. The link is there in the description. Thanks very much.